Good morning, student. Today I am going to discuss session 8 of chapter 6 from your class 12 biology syllabus. The name of the chapter is Molecular Basis of Inheritance. Already we have completed 7 sessions of chapter 6, and from those 7 sessions we came to know a lot of things about molecular biology. The first thing is different experiments on genetic material. The second, DNA structure and function. Third, nucleic acid structure and function. Fourth, DNA replication. Fifth, DNA transcription. And the last one is the genetic code and translation. So in this video we are going to discuss the gene expression in prokaryotic cell in meets the Lac Operon concept. So let's start the today's class. Today we are going to discuss Lac Operon concept or gene expression in prokaryotes or inducible operon concept. So we have to discuss all those things because all those things are very important for your exam. Okay, so this Lac Operon concept is very important concept, very important topic from your class 12 biology syllabus, and this topic is actually not important not only for your class 12 exam or board exam or any school exam. For the other competitive exam, this concept, this operon system, from that portion so many questions may come. Okay, so let's start to discuss. So actually this hypothesis for protein synthesis regulation in prokaryotes actually was given by Jacob and Monod. This Lac Operon concept was first proposed by French biologist Jacob and Monod. Okay, so there are two scientists. Just look at the picture of them, and experimentally they demonstrated the regulation of gene in E. coli cell. Okay, and for this experiment they were awarded with Nobel Prize in the year of 1965. So what is the meaning of this? That actually this hypothesis is known as the Operon model, and it was given on the basis of the study related to the enzyme beta galactosidase in E. coli. So as part of the DNA which actually act as the single regulated unit. So this operon model actually consists of the several parts. So first thing is actually this operon is a group of genes, and operon is a group of genes that are transcribed at the same time. I am repeating again, what is the meaning of the operon? So one operon, and operon is a group of genes which are actually transcribed at the same time. Okay, so just look at the picture. So these are the group of genes, and these genes transcribed at the same time at the same point. Okay, and produce different kinds of the product. It means the enzyme. Next thing is the sequential arrangement of the regulatory gene, the promoter gene, and the operator gene, and the structural gene in prokaryotes is called the operon concept. So what is the meaning of the operon concept, and how many parts are there actually? So this sequential arrangement, that regulatory gene is there, the promoter gene is there, the operator gene is there, and the structural gene is there. So by these four parts, one operon concept actually is produced in the prokaryotic cell. Okay, and each and every part has their different function. So these all the segments are actually part of the DNA which carry the codes for the synthesis of proteins, and these genes actually determine the primary structure of polypeptide chain by having the coding sequence of the amino acids in them. Okay, next thing is this Lac Operon consists of three structural genes. This Lac Operon actually consists of the three structural genes. Just look at the diagram. So it has total three structural genes, this LAC-Z, LAC-Y, and the LAC-A, and one of them actually gene for enzyme beta galactosidase. So one of these three genes, one gene produces the beta galactosidase, and the enzyme is the beta galactosidase, and the gene is the LAC-Z. The gene is the LAC-Z. Okay, so LAC-Z gene produces the beta galactosidase enzyme, and this enzyme hydrolyzes the lactose into glucose and galactose. Okay, so lactose transform into glucose and galactose. Lactose transform into glucose and galactose. Okay, so this is the overall structure of the Operon model. Next thing is the cultural situation for E. coli. So there are two cases. There are actually the two situation is there. The case 1 situation, that the when glucose is present and lactose is absent, the E. coli does not produces beta galactosidase enzyme. I am repeating again. So this is case study 1, that when glucose is present in the medium, okay, and the lactose is the absent, so that time E. coli does not produce beta galactosidase. And the case study 2, that when glucose is absent but the lactose is present, so that time E. coli produces beta galactosidase. Okay, so I am repeating these two case study because this is important for your exam. So when glucose is present but lactose is absent, so that time E. coli does not produce the beta galactosidase enzyme. And when glucose is absent and lactose is present, just vice versa. Okay, just opposite to of case study 1. So that time E. coli produce beta galactosidase. Okay. So the first point is the E. coli can use either glucose, which is a monosaccharide, or lactose, which is a disaccharide. Okay, so E. coli bacteria can use either glucose, which is a monosaccharide, or lactose, which is a disaccharide. So the lactose actually needs to be hydrolyzed. The lactose needs to be hydrolyzed. Hydrolyzed means the digested by the hydrolyzing enzyme and fast into glucose and galactose. So lactose actually needs to be break down into two components, that glucose and the galactose. Okay. Next thing is the some definitions are there. The first definition is the promoter gene. Okay, so this is the promoter gene. The PLAC is there, the orange color. The PLAC is there. So what is the meaning of the promoter gene? So actually it is located adjacent to the operator gene. So this red color structure is the operator gene. So it's actually situated or located adjacent to the operator gene, and it is actually the DNA segment where the RNA polymerase binds. So this promoter term, this term is actually, you have seen or you have noticed the DNA transcription procedure, because transcription procedure cannot start before the promoter region will be activated by the RNA polymerase. Okay, so it is the site for attachment of the RNA polymerase 2. It is the site of attachment of the RNA polymerase 2, and it promotes the structural genes to transcribe through the operator gene. Okay, so this is the site actually, this PLAC, this promoter site, actually the promoter gene, actually the site where the RNA polymerase binds. Okay, and it initiates the transcription of the structural gene. It actually initiates the whole transcription process of this structural gene. So this structural gene, so it controls the rate of mRNA synthesis, and the main site in promoter region of Lac Operon is the CRP site. Okay, so this is the promoter gene sequence. Next is the operator gene. So next adjacent site is the operator site. Okay, so what is the meaning of the operator gene? It operates and controls the expression of the structural gene. It operates and controls the expression of the structural gene. Okay, so this is a gene actually involved in controlling the expression of one or more other gene. So this operator gene actually controls or operates the expression of this structural gene, because this structural genes has to be transcribed. So these include the negative and the positive regulator. Okay. This next thing is the regulator gene. Next thing is the regulator gene. Okay, so what is the meaning of the regulator gene? So it's actually gene which is involved, which is control the expression on incorporation with the repressor chemical. So it regulates the operator gene. This regulatory gene actually operates the operator gene. Okay, controls the operator gene and incorporation with the repressor chemical. Okay. Last one is the structural gene. So these are actually structural genes are present next to the operator gene, and they are LAC-Z, LAC-Y and LAC-A. Okay. So this gene Z, gene Y and gene A. So gene Z actually contains 3063 base pairs and codes for the enzyme beta galactosidase, which breaks lactose into glucose and galactose. I am repeating again. So in Operon concept, in Lac Operon concept especially, there are three structural genes are there. Okay, so the name of these three genes are LAC-Z, LAC-Y and the LAC-A. So what is the main function of the LAC-Z gene? The main function of the LAC-Z that it contains 3063 base pairs and codes for the enzyme beta galactosidase. It actually codes the enzyme beta galactosidase. So just look at the structure. So this LAC-Z actually produce beta galactosidase enzyme and breaks. What is the main function of the beta galactosidase? The main function of the beta galactosidase that it breaks lactose into glucose and galactose. Okay, so during the time of your exam you have to remember, I am highlighting this portion, you have to remember the enzymes name properly. Okay, you have to remember these enzymes names properly, otherwise you will get simply 0. Okay, next thing is the LAC-Y. What is the this next thing is the LAC-Y. Okay, so I am clearing the picture first. So this is your LAC-Y. So what is the main function of the LAC-Y? So this contains 800 base pairs and codes for beta galactose permease. Okay, so this is the enzyme. The name of the enzyme is the lactose permease. Okay, or in the short form only permease. So what is the main function of it? That this LAC-Y gene codes for lactose permease. Okay, and contains 800 base pairs and codes for this enzyme, and this permease actually a membrane bound carrier protein which helps in the transfer of the metabolites. Okay, and the last gene is the LAC-A gene, and this LAC-A gene codes for the enzyme thiogalactosidase transacetylase. Okay, so it contains also 800 base pairs and codes for enzyme, this enzyme, and this what is the main function of this enzyme? The main function of this enzyme that this enzyme transfer the acetyl group from acetyl CoA to beta galactosidase. Okay, so during the time of your exam you have to remember and you have to mention properly all those enzymes name. Okay, so suppose if you forget all those things, then you will get simply 0, or if you write any kinds of wrong information, then you will get simply 0. So you have to remember all the enzymes name properly. So I am repeating again this structural gene portion because this is important. So this is actually the segment of DNA which carry the codes of the synthesis of protein. Okay, so these structural genes are present next to the operator gene. So there are total 3 structural genes are there: LAC-Z, LAC-Y and LAC-A. So LAC-Z gene actually codes enzyme beta galactosidase. I am highlighting this portion, beta galactosidase. Okay, and next thing LAC-Y, which actually codes for the lactose permease, and the next thing is the LAC-A, which actually codes for enzyme thiogalactosidase transacetylase. Okay, so each and every enzyme has their specific function. Each and every enzyme has their own specific function. Okay, so this is the actual structure of the operon. So this is next is the case study 1. Next is the case study 1. When inducer actually absent, the absence of the lactose. What happened in the absence of the lactose? Okay. So this is also called the switch off condition. So when the inducer is absent, so it means that when lactose is absent in the media, that time what happened, it activates the repressor protein, okay, with the operon gene, and it blocks it. So this repressor actually blocks the operon. So what happened after that, in this promoter site this RNA polymerase binds, but as because this protein, this complex, this repressor complex blocks this operon site, then what happened, then RNA polymerase cannot go to this structural gene, That's why this is called the switched off condition. Why? Because no transcription of mRNA is occurred by this structural gene. So structural gene cannot be transcribed without the RNA polymerase, and this RNA polymerase actually cannot go to this structural gene portion, cannot reach to the structural gene portion. Why? Because of this blockage. And how this blockage actually occur, that this regulatory gene actually produce the repressor protein, and this repressor protein directly binds with the operator site in the operon. Okay, so that's why this is called the switch off condition. So I am repeating again. So when actually the inducer is absent or its level falls off, suppose the lactose is present but their levels, actually the lactose concentration is very low, so that time they activate the repressor protein associated with the operator gene and it blocks it. So this repressor protein actually adds with this operator gene, so it forms another complex, and that time it blocks it. Okay, the whole site actually blocks. Okay, so after that the no transcription of mRNA is occurred by the structural gene can takes place, and thus there is no enzyme synthesis. Okay, so this is actually the structure that the E. coli bacteria that cultured in absence of lactose medium, it would not produces the enzyme beta galactosidase that necessary for the lactose metabolism. Okay, so if beta galactosidase is not produced properly, then what happen? Then lactose metabolism is also will be stopped, because beta galactosidase is actually the main thing which is necessary for the lactose metabolism. Okay, so in this condition structural genes are in switched off condition. The structural genes are switched off condition. Why? Because no transcription is occurred. And in absence of the lactose, the repressor protein synthesized by the regulator gene binds to the operator gene. So in absence of the lactose, the repressor protein actually binds with the operator gene and it blocks the site of the operon gene. Okay, and this blocks the RNA polymerase 2 to transcribe the structural gene, and hence enzymes are not produced. So if these three structural genes are not to be transcribed, then what happens? Then these three structural genes actually cannot produce any kinds of the enzyme. So that's why these enzymes are not produced. Okay, next, this is the case study 2. That in presence of the lactose, this is the switched on condition. Okay, so in the presence of the inducer, it means the lactose, in presence of the inducer, what happened? The repressor actually has a higher affinity for the inducer than that of the operator gene. So for that reason, actually when lactose is actually added to the E. coli culture medium, so that time a few lactose molecules are carried into the cells by the enzyme permease. Okay, and a small amount of this enzyme is present in the cell also there. Okay, so few lactose molecules are converted into the allolactose, and this allolactose actually binds with this repressor gene. So as usual, this operator site actually produces, this regulator site actually produces the repressor, but for the presence of the allolactose, this allolactose actually directly binds with the repressor and it forms another structure. It forms the one deformative structure. So that's why, so this whole complex cannot bind to the operator site. So for that reason, the promoter site, right now the promoter site is free. Okay, so RNA polymerase can bind easily with the promoter site and then it transcribes the whole structural gene and it produces the enzymes also respectively. The LZ, the Lac Z actually produces the beta galactosidase enzyme. Lac Y actually produces the permease and Lac A produces the trans-acetylase. Okay. So next thing is the E. coli bacteria that are cultured in presence of the lactose media, it produces the enzyme beta galactosidase. It is necessary for the lactose metabolism. It is necessary for lactose metabolism. Okay. Next thing is that in this condition, the structural genes are in switched on. Why? Because these structural genes are in transcribed mode. So these structural genes are become transcribed, okay, and produces the enzymes, respective enzymes. So that's why these structural genes are in switched on mode. And in presence of lactose, the repressor protein synthesized by regulator gene binds to lactose molecule and it forms the repressor induced complex. So this allolactose, actually this protein binds with this repressor gene and it forms one deformative structure. So for that reason, this complex become unable to block the operator site. Okay, for that reason the promoter site now free. So after binding the RNA polymerase 2 with the promoter, then it can easily reach to the structural gene portion and they can transcribe to produce the enzymes. So this is the basic things of the Lac Operon. So the inactive repressor complex does not bind to the operator gene. So the RNA polymerase to transcribe the structural genes to produce the enzyme. So this is the basic things of the Lac Operon. This is the basic things of the Lac Operon concept. Okay. So this Lac Operon concept is actually very much important for your exam. Okay, so this regulatory system of this gene expression is actually is very much important for your exam. Why? Because something is actually some tricky portions are also there in this topic, that the switch on and switch off mode. So this type of question may come from this portion, that when actually switched on mode is occur, why it's called the switched on, when the switch off mode is occurred, what is the meaning of the switched off mode, and what is the name of these all the enzymes. Okay, so which gene actually produce which enzyme, what is the main functions of each and every enzyme. So why this is called the switched on mode, and so in the switched on mode actually what happened. Okay, what is the basic difference between this switched on and switch off mode, what is the meaning of the inducer, what is the name of the inducer. So this type of tricky questions may come from this particular topic. Okay, so that's why go through these portions properly, because in today's video, from this video actually we come to know that what is the meaning of the operon, what is the meaning of the operon concept, and who discovered this thing, why it's called the operon concept, the parts of the operon concept. Okay, the structural genes, this promoter, this operator, this regulatory gene, all those things, how many structural genes are there, how many genes are there within the structural gene, what is the basic functions of this particular structural gene portion. And the next thing is the what is the meaning of this repressor protein. Okay, what is the name of the each and every enzymes, what is the particular function of this enzyme. So from this video we came to know a lots of things about the operon concept. So in our next class we will discuss the DNA fingerprinting procedure. Okay, so I hope this video will help you a lot for better understanding. I hope so, because this video actually contains this all those information, all those important information which is required for all of you, okay, which is needed for your board exam and for the other exams also. So that's why go through each and every videos properly and go through your NCERT books first and then follow each and every videos. I hope this video will help you a lot for better understanding. Okay, so now I am ending this session. So thank you and goodbye.